Okay, this is podcast number 2 for Standard 2.7. In this podcast, we're going to take a look at the process of meiosis. In meiosis, this is a special kind of cell division that is used solely for the formation of gametes. And at the end of meiosis, you're going to end up with 4 cells that are genetically different from each other and that are haploid. And this is very different from mitosis because in mitosis, you're going to get 2 cells that are identical to each other and are diploid. Problem is mitosis and meiosis sound very similar to each other, so a lot of people get confused. Meiosis basically is a double division process, so it happens in 2 steps. There is meiosis 1 and there is meiosis 2. So we're going to take a look at what happens during each process. A lot of the events are very similar to mitosis, but there are some key differences, and that's what we're going to highlight. So during meiosis 1, as with any kind of cell division, the cell has to duplicate its DNA before beginning. Remember, this happens during the S stage of the cell cycle, so the cell is going to make a complete copy of all of its DNA, both before mitosis and meiosis. And then this process in meiosis 1 is the first division. So we're going to be doing a division of the DNA twice in order to make haploid cells. So in this first division, we have 4 phases. And if you notice, the phases are pretty much the same as in mitosis. It's prophase, metaphase, anaphase, telophase. But these are all have the 1 after them because during the second division, we're going to go through these stages again. So let's take a look at what happens during this first part of meiosis. Prophase 1, a lot of what happens here is very similar to mitosis. So this is when the sister chromatids are going to become visible. This is when we can start to see our chromosomes that are in the X form. The nucleus is going to break down because we need to get those chromosomes out of the nucleus so we can move them around. The spindle fibers are going to form from the centrioles. Remember, the centrioles are going to move to opposite sides of the cell. And then they're going to start to make the spindle, and those fibers are what are going to attach to the chromosome so we can start to move them around. Okay, but then something else happens here that does not happen in mitosis, and that is a process where tetrads are going to form. And if you look at the word tetrads, you can see tetra there, and tetra means 4. So what we're going to actually end up doing here is we're going to end up pairing up these duplicated chromosomes that we have. So let's take a look at this tetrad piece of it. When tetrads form, we are pairing our duplicated chromosomes. So remember, our duplicated chromosomes are in this X form. They're made of a pair of sister chromatids. What we're going to do is we're going to take the homologous partner to that has already been duplicated as well, and they're going to actually get together very briefly. So what you end up with are 4 sister chromatids in this one little structure. So these are homologous chromosomes. Remember, they're similar to each other. They have the same genes on them and everything. And they're going to just very briefly kind of get together, kind of like they're holding hands for a minute. Now, during this time when they get together, an event called crossing over may happen. During crossing over, pieces of DNA can break off and trade places with pieces on other chromosomes. And basically what this can do is it can kind of shuffle the deck of cards. So we can kind of take our genes that are always on this one chromosome and kind of switch them up with a homologous chromosome. So now you don't have a chromosome that is identical to the one you got from your parents. You could be passing one on to your offspring that is different than one that you got from your parents. So we can take a closer look at this here. So in crossing over, here we have 2 homologous chromosomes. We know they're homologous because their genes are the same. These ones happen to have all dominant versions of the genes, that's why they're in capital letters, and these ones have lowercase versions of the genes, but this is still gene A, gene B, so they're going to do the same basic kind of job, just maybe in a slightly different way. So they are going to form tetrads, so they pair up with each other. Okay, so like here, they're actually crossing their legs around each other, so they're going to hold on to each other for a second. When that happens, sometimes little pieces break off. This is the crossing over event. So now some of those lowercase letters that we had on this chromosome are now going to be with the dominant ones. And so what we have here is when we end up separating these chromosomes during the process of meiosis, we're going to put them all into their separate places. Now we have this chromatid here, and we have one here that's different. We have this one here that's different, and this one here that's different. So we went from having what would have been 2 different kinds of chromatids when we separate them to having 4 different kinds of chromatids. And that's really important when it comes to genetic variation. So this little piece, this step in meiosis, is incredibly important when it comes to genetic variation, and why we all look a little bit different from each other. We don't all look exactly like our parents, because we actually shuffle our genes up quite a few times before passing them on to our offspring. Now, once the crossing over event has happened, we move into metaphase. And in metaphase, those tetrads, remember those are our groupings of our homologous chromosomes, are going to line up across the center of the cell. So just like in mitosis, where they line up, they're going to line up as well, but you can see they're still in their tetrad form, so they're still kind of paired up holding hands with their partner. In anaphase, we're going to pull those tetrads apart. So the tetrads are going to get split. The duplicated chromosomes are going to separate, the homologous chromosomes are going to separate, but they're still in their duplicated form. So they're still in their X form. Okay, so they're still copied. We don't want to give that copied form to our offspring. We need it to be separated. But right now, all we've done is we've taken our paired homologous chromosomes and we're just splitting them. So before they were together, kind of holding hands, right? And now we've taken them and we've made them two separate X's. And now they're going to be on separate sides of the cell, as you can see here. From here, we're going to, so remember, our homologous chromosomes are still in their sister chromatid pairs. So they're still in their duplicated form. They haven't been separated into their individual chromatids yet. And that's an important piece with this process of meiosis, because we're going to do a second round of division where we're going to get those down into their individual chromatids. Okay, so telophase 1 cytokinesis, this is the first division that's going to happen, and we're going to end up with two haploid daughter cells forming. So we had started out with a cell that had 4 chromosomes in it, and now these two cells only have 2 chromosomes in them. The problem is that they're still in their duplicated form. So because they're still in their duplicated form, we can't pass them on. We have to divide them again. Okay, so each cell now has 2 copies of the same set of chromosomes. We have 2 copies of this big chromosome, and we have 2 copies of this little chromosome. And again, you don't want to pass on 2 copies of the chromosome. You want to only pass on 1. So we have to do the second round of division. Now we're going to pause here for a minute, and we're going to do a quick comparison of mitosis and meiosis 1. So remember, during mitosis, the tetrads do not form. We don't do this little crossing over business that happens. That's important because we want all of our cells to become identical at the end of mitosis. And if this crossing over happened, then they wouldn't be identical to each other anymore. The sister chromatids are split during anaphase and mitosis, so that's when we're going to actually get our individual chromosomes. Whereas in anaphase 1 of meiosis, we are going to be keeping the sister chromatids together, but the homologous chromosomes separate. So at the end of meiosis, or at the end of mitosis, excuse me, the X form goes into 2 individual. But remember, during meiosis, it was in that X form, and all we're going to do is separate it into 2 separate X's. So we're pulling the X's apart, but they're still in that X form. Okay. At the end of mitosis, we're going to have 2 identical daughter cells, and those daughter cells are diploid. Okay, so they have a complete set of homologous chromosomes. They have everything that they're supposed to have. All of that, they have 2 copies of chromosome 1, 2 copies of chromosome 2. They have everything that they're supposed to have in them, and they're identical copies. Whereas at the end of meiosis 1, we now have 2 genetically different haploid cells. They're genetically different because that crossing over event happened. So all of the chromosomes are now no longer identical to each other. And they're haploid because we've taken the number of chromosomes and we've actually cut it in half, even though they're still in their duplicated form. So let's look at the second half of this and see if it becomes a little bit clearer as we move along. So in mitosis 2, this is the second division of meiosis, so we're going to now divide the cells again. So right now we have 2 cells, and we need to get them divided one more time, so we end up with 4 cells. So the sister chromatids are split, and they're going to be put into 4 different haploid cells. So now we're going to take all of our individual X's at this point, and we're going to make them into individual chromosomes that can be then put into the different cells. So we start with prophase 2. Remember, we now have 2 cells, but those chromosomes are still in their X form. Okay, so the spindles are going to attach to the sister chromatids again at the centromeres, just like in mitosis. And the nuclei that had formed temporarily are going to break down again, so we can move them around. Okay, so we have to start to kind of redo the process over again. Okay, but our end goal here needs to be to get these X's separated. Okay, so prophase, we're going to get reattached, the nucleus is going to go away, and then in metaphase, where now this looks a lot like metaphase of mitosis, those sister chromatids line up across the center of the cell. So that way we're now going to be able to separate these X's into individual chromosomes without them hitting up against each other and making it nice and easy to do. So they line up across the center of the cell, and now we are going to pull them apart. So the sister chromatids are now going to become individual chromosomes, and they're getting pulled away from each other to opposite sides of the cell. Okay, so this is, again, is very much the same as anaphase in mitosis. Once that happens and they get pulled to the opposite side of the cell, and again, this is happening in 2 cells at the same time, then we move into telophase 2 and another round of cytokinesis. And we're going to get 4 new nuclei formed. So each of these cells that have a chromosome in them are now going to get a new nucleus going around each set of chromosomes, because chromosomes like to be inside a nucleus. The cells are going to divide and split into 4 cells. And what we end up with are 4 different haploid cells. Okay, they're haploid because if you look, there's now only 2 chromosomes, and our original had 4. They are different from each other because if you look at the chromosome, they've been mixed up with their color. So here you have got like a small one that's all blue. Well, here you have a small one that's mostly blue with a little bit of green, right? Here's a small one that's all green. Here's one that's mostly green, a little bit of blue. If you look and compare these cells, all 4 of them have different variations of these chromosomes, so none of them are identical to each other. Okay. So each cell now has 1 copy of each kind of chromosome. So when these cells go to get passed on to the offspring, it's going to give 1 short one and 1 long one. The other short and long one are going to come from the other parent during the process of fertilization, and they will pair up. And again, we have done some shuffling of genes here, so we've got a lot of variation that's happening. These chromosomes, like this chromosome right here that's green in the middle with blue ends, is not at all the chromosome that this organism had received from its parents. It's now been mixed up and jumbled up together. So it's a mixture of those 2 parents, and that's what's going to get passed on to the offspring. And this is how we get genetic variation. Okay. So in humans, at the end of this process, each of these new gametes that we have will have 23 chromosomes in them. And that is our haploid number, and that's what we want to pass on to our offspring. All right, so one of the things that NCAS people like to have you do is to compare mitosis and meiosis. And so we kind of look at the things that are pretty different about them. That's usually the easiest way to look at it. So if we look at mitosis, remember mitosis is used for the formation of somatic cells during growth and repair and things like that, and also can be used for asexual reproduction of certain unicellular organisms. Your cells are going to divide only once during mitosis. And in the end, you're going to get 2 identical daughter cells. Okay, so those cells are going to be copies of each other, copies of the parent. And those cells are going to be diploid. So they're going to have a complete set of homologous chromosomes. They're going to have 2 copies of everything that they're supposed to have. 2 of chromosome 1, 2 of chromosome 2. They're diploid. That's what most of your cells are like in your body. They undergo mitosis. They are the ones that determine everything about you. On the flip side of that, if you look at meiosis, meiosis is for the formation of gametes and the formation of gametes only. The cell is going to go through division twice. And in the end, because the cell is dividing twice, you're going to end up with 4 cells that are genetically different from each other. Remember that genetic difference comes from the crossing over event. Okay. And the cells that you're going to get at the end are going to be haploid cells. So they have half the number of chromosomes as your regular somatic cells. Okay, so your somatic cells in your body have 46. These guys are going to have 23 because that's what you're going to pass on to your offspring. Now the big major difference between meiosis and mitosis is this formation of the tetrads and the crossing over. The crossing over is going to give us greater genetic variation, and that greater genetic variation is going to be really good for the species. I talked about that before, that with more genetic variation, the species is more likely to survive changes in the environment. Okay. Okay, so remember, genetic variations are the differences in genetic makeup among members of a species. So if you look around your class, you can see that, we all don't look the same. There's a lot of different characteristics that are in there, and that happens because of this process of crossing over that occurs and sexual reproduction. All right, so we can take a look at this and just kind of go through this whole process and see how we would develop a multicellular organism. Okay, so you need both mitosis and meiosis to make a multicellular organism. The first step is the gametes are going to be made by meiosis, so we have to form the gametes first. The haploid male and female gametes, the sperm and the egg, are going to come together by the process of fertilization, and they're going to make the zygote. Okay, so we take our two haploids cells and come together and make a diploid zygote. That zygote is going to become a multicellular organism by undergoing mitosis many times. So remember, I showed you that zygote, that fertilized egg, is going to divide. That's what's going on during pregnancy, during development in the womb, is that cell is dividing over and over again until it becomes trillions and trillions of cells. Okay, and remember that organisms grow by adding on new cells through the process of mitosis. You want your cells to all have the same DNA in them. You want them to be identical, so that's what's going to happen. Okay, and this just kind of shows you how the process can happen. So we could start here with meiosis. Meiosis is going to make those gametes. The gametes do fertilization. Those are haploid cells on that side. Fertilization is going to make our zygote, which is diploid. And then that zygote's going to go undergo mitosis a whole lot until you make your multi-celled body. That multi-celled body is then capable of undergoing meiosis, and the process just keeps continuing over and over and over again.